So now let's specifically talk about those individual organelles we find. Well, when I was describing common characteristics you see between cell types, I mentioned an enclosed nucleus or a nucleus surrounded by a membrane. But let's go over what a nucleus is. Well, it says here the nucleus contains most of the genes in the eukaryotic cell. So the nucleus is you can kind of think of it as like the heart of the cell. It's where all the genetic material is going to be contained. And here it says in the second bullet, it says the nucleus has something called a nuclear envelope, which encloses the nucleus, separating its contents from the cytoplasm. And remember that cytoplasm was all that kind of liquid goo within the cell that's separating all the organelles from one another. Okay, it's kind of like that fluid within the cell. So the nuclear envelope separates the nucleus from that cytoplasm. And then, since all of our genetic material is found within the nucleus, we organize that genetic material, which we call DNA, into these structures called chromosomes. And you've probably heard of a chromosome before, but maybe you didn't know specifically what it was. Here it says chromosomes are discrete units of organized DNA. So it's our body's way of organizing DNA and kind of containing it within an organelle. And again, all of this information is found within the cell's nucleus. Now, to get a little bit more specific, if we're talking about genetic material or DNA, there's this substance they call chromatin, okay, and it's the material that makes up chromosomes. And simply put, chromatin is just kind of a mixture of that DNA we talked about as well as the proteins. So in order to have chromosomes, we have to take proteins and DNA, and we combine them into this substance they call chromatin. That chromatin then comes together to create chromosomes. Now, at the very, very center of our nucleus, we have something called the nucleolus. And we'll talk about this more later in this course, but the nucleolus is an area of the nucleus where we find this material called rRNA. And I think when we talk about genetic material, we typically think of DNA, but there's actually another type of nucleic acid that we call RNA. And this is where we're going to find a lot of it. And when we start talking about cell replication and DNA replication, we'll talk more about RNA. But for right now, I just want you to be familiar with the fact that the nucleolus is located within the center of the nucleus, and it can contain RNA. So this is just kind of a visual representation of the nucleus and a lot of the different components, and there's more information here than you need to know for this class, but I like to have some visuals. So again, we have kind of a character illustration of the nucleus, and it's that purple blob in the upper right-hand part of your screen, but you can see in the middle of it, again, we have that nucleolus, which contains RNA, and then within just the rest of the nucleus, we have these kind of string-looking features. And that's actually the chromatin, or our genetic material, such as our DNA. And then on the left-hand side of the screen, you can see just kind of an actual photograph of a nucleus. Now, ribosomes are extremely important for cells, and ribosomes are particles that are made up of rRNA and proteins. So we mentioned that we find RNA within the middle of a nucleolus, which we find in a nucleus. But ribosomes are really important because they help our body carry out protein synthesis. And when we talked about macromolecules, we talked about how important proteins are. They function in so many different, levels all over our body. I mean, they participate in every single thing our body does. So we need to be able to create proteins, and we can do that because of these organelles we call ribosomes. Now, there's two types of ribosomes I want you to be familiar with, and those are free and bound ribosomes. And really, they're named as such because of where they're found within the cell. So it says here, free ribosomes are suspended in the cytosol. So if you have free ribosomes, they're simply going to be kind of floating around in that cytoplasm, in that liquid goo within our cell. However, if we have bound ribosomes, those are going to be attached to something. And it says here, bound ribosomes are attached to the outside of the endoplasmic reticulum or nuclear envelope. So if we have a bound ribosome, they're going to be attached to some other structure. And typically they're attached to this organelle they're calling the endoplasmic reticulum, which we'll talk about in just a minute. So here's just another illustration for you. On either side, you have kind of the character drawings, if you will. And then within the middle, you have a picture of ribosomes. And so as you see, you can kind of see these three distinct lines that are running through the photographic image. And those are our bound ribosomes that are attached to parts of an endoplasmic reticulum. So remember, if they're attached to some other substance, if our ribosomes are attached to some other substance, we're going to call those bound ribosomes. However, in the spots between those three lines that are running through the picture, we see just black dots in kind of a fluid-filled space. And those are our free ribosomes, or our ribosomes that are kind of located within the cytoplasm or the cytosol. Now, what in the world is this endoplasmic reticulum we've mentioned? Well, the endoplasmic reticulum consists of a network of membranous tubules and sacs that they call cisternae. And there's two distinct regions within our endoplasmic reticulum. And sometimes people just refer to this as the ER, just because you don't have to say that long, crazy name. But, again, because it works with ribosomes, we're going to have two different types of endoplasmic reticulum based off of whether or not they have ribosomes attached to them. So if we have smooth endoplasmic reticulum, or if we have a smooth ER, that's simply going to mean that it's endoplasmic reticulum that does not have ribosomes attached to it. If we have rough ER, that means that we're going to have a structure that's going to have ribosomes attached to it. And this is an illustration of the endoplasmic reticulum, and you can see how it has a lot of these kind of folds and curves to it. And you can see that on certain spots of our ER, we have little red dots, and those little red dots, again, are those ribosomes that are going to assist with protein synthesis. So, what are the functions of the endoplasmic reticulum? Well, it differs slightly depending on whether you're talking about smooth ER or rough ER. But here there's 4 examples of what a smooth ER is there for. What is its purpose? Well, this first bullet says it's able to synthesize lipids. And if you recall from our macromolecule lecture, lipids are fats. So we need to be able to create fats for different parts of our body. And we're able to do that because of this smooth ER. Another function it does for us is it metabolizes carbohydrates. So that's really important, right? We like to eat our carbs. So we need to break them down somehow, and our smooth ER is going to help us to do that. Smooth ER is also helpful in that it allows us to store calcium, and it can also help us to detoxify poison. What's really cool is they've shown that different types or different ERs within the human body can actually double in size if somebody ingests poison, or if maybe you drink too much alcohol or something like that. It actually gets larger to help compensate for all that poison it has to detoxify. Now, the rough ER was the other type of endoplasmic reticulum, and it has a slightly different function. Well, it's rough, which means it has ribosomes, so it has ribosomes bound to it, and it's going to produce proteins and membranes, which are going to be distributed by different types of transport vesicles we'll talk about. So that would make sense if we think about the fact that ribosomes are there to help produce different types of protein, and if they're attached to the rough ER, it's also going to assist with the production of proteins. Now, this next organelle we're going to talk about is called the Golgi apparatus, and this is known as the UPS center of the cell. So if you didn't read anything else, you could probably guess at what this organelle is going to do for the cell. If it's the UPS center, it's going to be receiving and delivering different packages or different products. Well, let's read this first bullet. It says, after leaving the ER, many transport vesicles travel to the Golgi apparatus. And here it says the Golgi apparatus is a center of manufacturing, warehousing, sorting, and shipping. So basically, different materials are going to come into this Golgi apparatus that our body has either taken in or produced, and the Golgi apparatus is going to package it in such a way that it can then ship it off so that it can be used by other parts of the body, or it can be used within other parts of the cell. So here, products of the ER are modified and stored and then sent to other destinations. And it says here the Golgi is specialized for secretion, and we'll talk about why that's important in just a minute. Now, if we're receiving products within the Golgi apparatus and we're also sending off products, we have to have two sides, or two poles, they like to call it. So we have one side that they call the cis face, and then on the other side of our Golgi apparatus, we have this side that they call the trans face. Now, the cis face is located near that endoplasmic reticulum. Okay, so it was located near that structure that often had ribosomes attached to it. And what happens is that the cis face is the side of the Golgi apparatus that receives products. So here it says, vesicles from the endoplasmic reticulum can move to the Golgi apparatus. And these products, or these vesicles, are going to fuse to the cis face of our Golgi, or of our Golgi apparatus. So what do I want you to grasp from this? I want you to be able to tell me that the part of the Golgi apparatus that receives materials is called the cis face. Reversely, I want you to be able to tell me that the part of the Golgi apparatus that kind of ships off products is called the trans face. So this final bullet says, the trans face gives rise to vesicles which pinch off and travel to other sites. And here's another illustration for you again. On the left-hand side, you can see, if you look at that bulleted number 1, it says vesicles move from the endoplasmic reticulum to the Golgi apparatus. So you can see that they're fusing to one side of the Golgi. And here, we would call that, because we're receiving products, we would call that the cis face. Now, if you move to the other side of the Golgi apparatus, you can see that there's little pieces that are going to be getting pushed off. Okay, and that's because they're being shipped out to other parts of the cell. So we're going to call that the trans face of the Golgi apparatus.